This unit, we're going to discuss staining processes and using different types of stains and their methods of examination, along with some basic tips on microscopy. Please review these objectives on this unit. This slide shows a diagram of a basic bright field microscope. This type of microscopy is what is typically used in the microbiology lab. We primarily use this for doing our gram stains, but it can be used for any other type of direct microscopy, some direct stains, anything really other than your fluorescent stains. And just pay attention to the particular terms on the left. I trust that you are familiar with how to use a bright field microscope, but in the microbiology lab, you're primarily going to be using all your different objectives. So you should have both low power fields, mid-range, and then oil immersion, high power objectives as well. Be familiar with how to adjust the amount of light, both with the condenser, being able to lower the condenser, as well as the diaphragm, how much light you're going to be letting through, and then just the ability of understanding how to clean and prepare a microscope for use. There are different direct examination methods, but honestly, we don't use too many of these anymore in a microbiology lab. But they are listed here. We'll go through some of them. A saline mount is obviously the most basic type of direct examination you can do. And primarily you might use this to distinguish between whether or not you have the presence of bacteria versus yeast. And you would primarily want to really lower the condenser on this, since you don't have any stain that will distinguish any features. But that we do occasionally use a saline mount. Iodine mounts are primarily used in ova and parasite examinations. Many clinical microlabs may no longer do a full ova and parasite examination. Most of these specimens for O&P are going to be sent to a reference lab where they would probably do an iodine mount. But in case you are in a lab that does do the full ova and parasite examination, that iodine mount is useful to the iodine itself will be taken into the cells of parasites and allow for the ability to see different parts of the intracellular items of within the parasites themselves, both cysts and trophozoites. And once again, that typically is done, and we'll talk more about that when we talk about GI pathogens. Potassium hydroxide, or KOH, is a preparation that might be used to look at direct specimens for fungal elements. This is particularly useful looking at skin, hair, and nails. The type of fungus called the dermatophytes are infections, particularly of in these parts of the body, skin, hair, and nails. And typically in most microbiology labs, they might use the KOH as part of a fluorescent stain that gives a even better highlights of the different parts of the fungus. But it's possible that the KOH alone might be used, even sometimes in the emergency room. Physicians, particularly if they are dermatologists and are familiar with what they're looking for, might do direct smears if they have access to both the reagent and to a microscope. India ink. This is a stain that is used to look for the capsules of a cryptococcus, which is a yeast, and a serious infection that can cause meningitis in patients, particularly who are immunocompromised. It's only about 50% sensitive, so along with all of these direct examination methods, they are not particularly sensitive and also not necessarily specific. But it does highlight the capsules which will not stain around those yeasts. So when we do have a positive India ink, it's actually pretty cool to see. But once again, other methods for looking for Cryptococcus are preferred over the India ink. And then there is the Neufeld or quelling reaction, which allows to look for bacterial capsules or the capsular swellings for bacterial capsules. So once again, here's a list of these direct examination methods, and it's helpful to know at least a basic idea of these methods, even if you may not see them while you're doing your clinicals. This slide, it contains a few links to preparations of different types of smears. It may be helpful for you to click on and watch these YouTube videos. And there's a difference between simple stains and differential stains. It's possible to do a simple stain is basically going to allow you to look at the shape, the size, and the morphology. This example here shows a methylene blue stain. Methylene blue is just one particular stain. It will allow you to, once again, just look for morphology, and but doesn't allow you to differentiate between different types of bacteria. It can be helpful just looking for the presence of, for instance, polymorphonuclear cells within stool specimens. That's one method sometimes of looking for fecal leukocytes would be just by staining with a methylene blue stain. Whereas differential stains, such as the Gram stain, our primary stain in the microbiology lab, that allows you to look for those same size, shape, and morphology, but also the Gram reaction, which is very significant, is our primary way to make our first distinction in bacteria. So obviously, you know, I like to talk about junk in gives you junk out. So starting with the preparation of a smear is what allows you to have the beginnings of a good end of your staining. So you would apply the specimen to a slide, and it depends on what kind of specimen you receive in the lab. If you have a liquid specimen, you will typically put a drop of that liquid onto a slide. If you have a swab, then you would roll that swab onto a slide. And you could use that with either a swab or a wood applicator stick. But the most important thing is to make sure that you have a very thin and clearly defined specimen on the slide. If it is too thick, number one, much of the specimen can wash off in the process of staining, and so that could either lead you to think that you don't have any bacteria present, but it's because the specimen has been washed off. Or it's just so thick, it's hard to get, either you might have a lot of stain precipitate, which could mask the presence of bacteria, or it just might change the color. You may not get as good decolorization or your counter stain. So what you really want is a monocellular layer. So you really, after you apply the specimen, you want to make sure that that is spread out nice and even. And then, of course, you need to fix these slides. You can do that either by heat or by methanol. And so if it's methanol, you're going to let that air dry, and then the heat obviously would typically go on a slide warmer. And the Gram stain, and we'll talk about this in more detail in the following slides, you have four stains. Your primary stain, crystal violet, your mordant, iodine, your decolorizer, either an alcohol or acetone, and your counter stain is safranin. And we'll talk about that a bit more. So the first thing we're going to do is talk about the morphology of the bacteria that we're going to be looking at in these Gram stains. We would first divide them by their shape. So cocci, which would be round or spheroid, bacilli, which are going to be rod-shaped, and spirochetes, which are going to be in spirals, kind of twisted. So cocci, typically you're going to see them in three distinct kind of formations. So one would be in pairs, where you would have two cocci that are sort of formed next to each other, and they are formed as if almost in one unit of two. And sometimes they're going to be two perfectly round cocci. Sometimes they'll be a little bit more elongated or lancet shaped, we would call them. And sometimes they're kind of in between a cocci and a bacilli, a little bit rounder but stretched out a little bit. Okay? And there are certain organisms that are associated with these different shapes, which we'll talk a bit more about later. And when we refer to these cocci in pairs, sometimes you might see them referred to as diplococci, meaning two. Then you would have cocci that are in chains. Okay? That means there are more than two that are connected, sometimes many, many more than you might have, 6, 8, 10 that are all sort of connected. Okay? And the type of bacteria that this typically represents would be our streptococci, and that is typical. Those are typically in chains. The last type of cocci would be in clusters. So almost think about a bunch of grapes. Okay, they would be in this clump or cluster of cocci. And typically we're looking at staphylococci, and these organisms we're going to talk about in the next unit or two. And we talk about bacilli. Once again, these are rod-shaped, and these rod shapes can be different lengths, and the way they are formed is sometimes they are square-ended, sometimes round-ended. They can be fusiforms, which means they're going to be long and maybe tapered at the end. They can be very filamentous, which also they're going to be kind of long and sometimes branching a little bit. Once again, when we talk a little bit about, I mentioned this with the cocci in pairs, there can be coccobacilli, which means they're still bacilli, but they're they're shorter. Okay, they're going to be kind of in between a cocci and a bacilli. Okay. And sometimes even you might see some of these bacilli might even have some chaining. Typically your spirochetes, which honestly we don't see these often in gram stain. Spirochetes tend to not stain, and we typically look for them using other methods like immunology and serologies. Okay. Once again, all of these different morphologies, particularly of the cocci, are seen best in a direct specimen or grown in a broth. If you were staining directly from a colony, you may not get a nice group of chains or clusters. Everything might look a little bit like it's kind of clumped together. So we're really talking about the best way to describe the morphology is in a direct stain of a specimen or once it's grown in a broth. Okay, let's delve a little bit more into the actual gram stain. There are 4 reagents I mentioned before. The crystal violet is your primary stain. And just backing up a minute, when we're talking about stains, particularly when there's more than one reagent, you would typically have a primary stain, some type of decolorizer of that primary stain, and then a counter stain, which would, if you get rid of your primary stain, the counter stain would give you some color to look at. So primary, decolorizer, counter stain. So for gram stain, our primary stain is crystal violet, and it will stain all bacteria dark blue to purple. We have a second stain called Graham's iodine, and that acts as a mordant. Okay, that enhances the reaction between the cell wall and the primary stain. So if you think about, it just kind of helps bind that primary stain to the cell wall. Then your decolorizer, and depending on the lab that you're working in, it's either going to be acetone or an alcohol, ethyl alcohol, or maybe even a combination, something that's a little weaker than acetone's pretty strong. Sometimes you have a combination acid alcohol. And at this point in the decoloration, a gram-positive bacteria, that's going to be one that will ultimately stain purple. The gram-positive bacteria will retain the primary stain, and we're going to get more into why that is. Okay, it's because of the peptidoglycan in the cell wall, but we'll get into that. The gram-negative bacilli loses the primary stain because of a large amount of lipopolysaccharides. Okay. Once again, we're going to jump into that a little bit more. And then your counter stain is typically safranin, and it will have no effect on the gram-positive bacteria because they never lost their color of the crystal violet, okay? But it allows for the staining of your gram-negative bacteria, and they're going to be pink to red. Okay, I love a good graphic. I'm a visual person. I like to kind of follow along and kind of see what I'm looking at. Okay, this is the really basic procedure for the gram stain. Okay, the procedures on the right and on the left is what it looks like at every step. Okay, so the first step, once again, we mentioned this, is for all of our stain preps is to do some kind of fixation. So the material is fixed on the slide, and of course it's cooled to touch before staining. Then we flood the stain with crystal violet, which of course is purple, and it stays on the slide for 1 minute. So everything, both your gram-positive and your gram-negative organisms, are going to stain purple. Then you rinse it with tap water. Then you flood the slide with mordant, which is your iodine, and that stays on the slide for 1 minute. And that, once again, that mordant binds the stain to the cell wall. At that point, you're going to, after 1 minute, you're going to rinse that again with tap water, and now you're going to add your decolorizer. It says flood the slide, but really what you're doing is you're just allowing that decolorizer to run down the slide 1 to 5 seconds. And it really depends on what type of decolorizer you're using. Acetone's very strong. 1 or 2 seconds is plenty. If you're using a little weaker decolorizer, such as your alcohols, those you might need 5, 6, 7 seconds. Okay. So at this point, and you'll notice in the diagram, the image, that your gram-positive organism is still going to stay that dark purple. The decolorizer is going to do nothing to that. But your gram-negative organism has lost the ability to hold that crystal violet during the process of decolorizing. So you flood the slide with a counter stain, safranin, for 30 seconds, rinse and dry the slide. So now you're going to see your end result is that a gram-positive organism is going to stay that dark purple, and your gram-negative organism is going to be a pink variety, either a light pink, dark pink. Different organisms will have a little different color of that pink. Okay, exercise 1. Be prepared to talk about the gram stain procedure and how this color would, what would happen after the iodine step. What would the color of the gram-positive and gram-negative organisms show? This slide is an example of gram-positive bacteria versus gram-negative bacteria. You can clearly see the purple staining of the organisms on the left and the pink staining organisms on the right. This is an example of a gram-stained smear. This is Streptococcus, so you can actually see those nice chaining organisms. Some of them are shorter, but many long chains. And this is following the growth in a broth culture. Sometimes you'll see the morphology of your gram-positive cocci. They tend to form the pairs, chains, and clusters. You can see that more clearly out of a broth or a direct smear than from the individual indirect colony gram stain. The organisms may be aging because they do appear slightly gram variable, but you can still see the purple in them. And then the chaining is very much indicative of a Streptococcus. So we're going to discuss gram-positive organisms. The gram-positive cells, they take up the crystal violet which is then fixed in the cell with the mordant, iodine. We discussed this when we talked just about the process of gram staining. So this mordant, this crystal violet iodine complex, it remains in the cell even after decolorizing. And there's some things about the cell wall structures of gram-positive organisms that people think that actually kind of make this happen or cause this complex. The cell wall of gram-positive organisms includes a thick layer of a protein sugar called peptidoglycans, that protein sugar complex. And it makes up the majority of the gram-positive cell wall. So the decolorizing of the cell causes this thick cell wall to dehydrate and shrink, and that actually closes the pores in the cell wall, and it prevents the stain from exiting. So it even forms a tighter complex. At the end of the gram staining procedure, then, the gram-positive cells will be stained a purplish blue color. Gram-negative organisms, the cells equally take up the crystal violet, just like the gram-positives do, and the iodine does form a complex with the crystal violet, just as it did with the gram-positive cells. And peptidoglycan is present in the cell walls of gram-negative organisms, but not to the extent as the gram-positive. So only 10 to 20% of the cell wall. Gram-negative cells, they have an outer layer, an outer membrane or outer cell membrane, which the gram-positive organisms don't have. And this layer is made up of lipids or fats, polysaccharides, and proteins. So the exposing of the gram-negative cells to the decolorizer, it dissolves the lipids in the cell wall. So now that crystal violet iodine leaches out of the cells. And now the cells can be stained with the safranin because they don't have the same, that complex is no longer the same structure like the gram-positive cells have. So now at the end of the gram staining procedure, gram-negative bacteria will stain a reddish pink color. Exercise 2. Please be prepared to discuss. We're going to discuss some other stains that are used in the microbiology lab to stain other types of bacteria. Acid-fast stains are used to stain acid-fast bacilli. Those are bacteria whose cell walls possess thick, waxy cell walls. They contain a large amount of mycolic acid. Once these organisms are stained, the bacteria are resistant to decolorization, typical decolorization, even by acid alcohol and acetone. So now they're called acid-fast because they are resistant to the acid in the decolorization process. And most acid-fast bacilli are known as Mycobacterium. Thus, Mycobacteria tuberculosis, avium complex, fortuitum. These are several species within the Mycobacterium genus. So the classic stain of for acid-fast is the Ziehl-Neelsen technique. It utilizes carbofuscin. And which is it's a very deep red, deep pink stain. And in the Ziehl-Neelsen technique, the carbofuscin is heated to drive the stain into the cell wall of the Mycobacterium. This is not, it's no longer used often in a routine microbiology lab. Heating of the carbofuscin is not the most practical. So now it's much more typical to use the Kenyan carbofuscin method. That stain has a higher concentration of phenol, and that enables the stain to penetrate that cell wall without the heat. In this case, the acid-fast bacteria stain red, and then the counter stains of methylene blue are the kind of the debris in the background. So thus, this photo on this slide shows an example of a Kenyan stain. The bacteria, mycobacteria, are staining pink, and then the background cell structures and debris stain blue. Please follow the link to view that procedure. So here are some examples of fluorescent stains. I'm going to start with the rhodamine because that is the staining method for Mycobacterium, which we discussed just previously. So the rhodamine stain, it's a fluorochrome used to stain Mycobacterium, and it will stain the bacteria fluorescent yellow to orange with a very dark background. So you can see here it's almost black, and then very bright fluorescent orange bacilli, which you can clearly see. Now the acridine orange, which is at the top right, is an interesting stain. It actually selectively binds to any nucleic acid. So it's particularly useful when to demonstrate small amounts of bacteria, particularly in blood cultures or spinal fluids in which you are suspecting there to be organism but are unable to see them on a routine gram stain. Particularly if a blood culture goes positive on the instrumentation and but we are unable to see organisms growing. Acridine orange is a good follow-up to that, just to see if there's any nucleic acid that is staining. It is non-specific, so it will bind to any nucleic acid, including host cells. And then the bottom right stain, it's my favorite fluorescent stain, calcifer white. I really like yeast and fungi. The calcifer white is used to stain specimens for the presence of fungi or fungal elements. They appear bluish white with a fluorescent microscope, and they just stain much better than using light microscopy, and you can much more sensitive. All of the fluorescent stains really are very sensitive. You can stain and pick up very small numbers of organisms because of the fluorescent, the organisms will jump out and much easier to see. Okay, we're going to look at a few other fungal stains that are not fluorescent stains. Lactophenol cotton blue is a very common stain in the microbiology lab, and it's used to stain fungal structures. So it is the lactophenol cotton blue is made up of lactic acid, and that preserves the fungal structures. And phenol, which actually kills any living fungus, and cotton blue. And the cotton blue stains the chitin in the fungal cell walls an intense blue color. So thus you can see, this is an example of an Aspergillus species, and you can see the structures very well. Then in the methenamine silver stain, it's really more of a histological stain. So you would see it in the pathology lab, typically would use this stain. And it stains fungal elements primarily that used to stain tissue samples. And then the PAS stain is used to stain molecules with a high carbohydrate content, like glycogen, and is generally preferred for staining direct clinical specimens. I would say that is used less frequently. Typically, you're going to see the MSS stain and in the histology lab, and then the lactophenol cotton blue to stain your fungal structures once they've grown, and then the calcifer white to do direct specimen staining. So there's also antibody conjugated stains. These are less frequently seen in a routine microbiology lab. But you can detect antigens of a corresponding organism by binding specific antibodies to stain. And then immunofluorescence uses an antibody conjugated to a fluorescent dye. So then you can use it for direct examination of patient specimens, And you can do direct immunofluorescent techniques can be used to detect Legionella and Bordetella in clinical specimens. And then enzyme conjugated stains, such as horseradish peroxidase, and it actually produces a color end product and can be measured photometrically. It's primarily used to detect viruses. But once again, as I said, we don't typically see this in a routine microbiology lab, but it is out there. This is exercise 3. Please review, be ready to discuss. And this is the end of this unit.